Welcome to UC Grad Slam 2022. I'm Katie Ferrick from LinkedIn. Welcome also to LinkedIn San Francisco. LinkedIn is the world's largest online professional network. 6 people are hired every minute on LinkedIn from our community of 830 million members. 50 million people use LinkedIn each week to search for jobs. We aspire to create a trusted, caring, inclusive, fun, and transformational experience for each other and through our platform for our members. As we endeavor to transform our world of work, innovation plays a critical part. And that's why we are so excited to welcome back the UC Grad Slam for 2022. The research represented here is an excellent indicator of what is new and what is yet to come. The University of California system plays a critical part in our innovation economy in our state, in our nation, and around the world. You've all learned, discovered, and distilled your research findings in a way that will help us all dream big. So let's get this started. It's my great honor to introduce the leader of our phenomenal UC system, Dr. Michael V. Drake, MD, President of the University of California. Dr. Drake was appointed as the 21st President of the University of California in August of 2020. He oversees the UC's world-renowned system of 10 campuses, 5 medical centers, 3 nationally affiliated labs, and more than 290,000 students, 230,000 faculty and staff. Dr. Drake has led the Ohio State University from 2014 to 2020, and prior to OSU, he spent his entire academic career at the University of California, including 9 years as Chancellor of UC Irvine and 5 years as the system-wide Vice President for Health Affairs. So please join me in giving a warm welcome to President Drake.
Well, thank you very much, Katie. Good to see you and great to see everyone here today. This is a special and exciting morning for us. Welcome to the University of California Grad Slam competition. How about a round of applause for that? And I'm very pleased to be here today with some of our university's brightest minds. It's a special experience to be back in person for Grad Slam, and given the pandemic, we continue to take precautions, so we don't have a general audience with us today, but we're very excited to be gathering in person. And first and foremost, I want to begin by thanking our gracious host, LinkedIn, for once again providing space and support for Grad Slam. We're grateful for your partnership and helping the University of California make this event such a success. So thank you so much for that. Grad Slam is a joyful celebration of one of the most fundamental missions of the University of California, the pursuit of new knowledge, and the sharing of that knowledge and those discoveries with the world. Graduate students play a critical role in the daily work of research and discovery, and it's thrilling to hear directly from 10 of them this morning. The 10 graduate students we are about to meet are already champions at explaining their work and their research in an engaging, dynamic, and understandable fashion. They have mastered the art of simplifying and explaining complicated research to anyone, and that means everyone. So everyone watching today should prepare to have a good time hearing about some of the fascinating work that our students have been doing. Before I tell you about our 5 distinguished judges, I'd like to take a moment to thank Thermo Fisher Scientific for being here today. They have given generously and they've sponsored all of today's Grad Slam cash awards. That's another applause line, I think. We're thrilled to have them as a part of Grad Slam and thank them for the steadfast support over the years. Now let's meet our panel of judges. I'd like to read a brief bio of each of these outstanding leaders, and please feel free to refer to the judge bios in the electronic program as well. Well, we have a great group of judges this morning. I'll begin with Jose Hernandez. Regent Hernandez is a UC Regent and is president and CEO at Tierra Luna Engineering, LLC, an aerospace consulting company. Mr. Hernandez worked at NASA Johnson Space Center from 2001 to 2011, where he was an astronaut. You know, I was reading the bio, it says he was an astronaut, then a bunch of other things. I think I just thought, well, he was an astronaut. And I got in a rocket ship and they shot me into the space. Okay, I got it. That was enough. But prior to that, Mr. Arnett has worked for 17 years at Lawrence Livermore National Laboratory, where he worked on the X-ray laser project, co-developed the first full-field digital mammography system, and also worked in the nuclear non-proliferation arena. The next judge is Nicole Estrada. Nicole is a senior at Gateway High School in San Francisco. She will attend UC Merced for the fall, majoring in public health with a minor in psychology. She has also served on the student council since her junior year and is now the student council treasurer. As a community leader for the Latinx Student Union, Nicole has been an important part of developing and maintaining the Latinx community, especially through the pandemic. She is a student athlete and is captain of varsity soccer. Nicole. Our third judge is Alan Sachs. Alan is the Chief Medical Officer for Thermo Fisher Scientific. Dr. Sachs' efforts include identifying, developing, and commercializing products and services that enable clinical customers and in leading the medical affairs program. He served as the Chief Scientific Officer for Thermo Fisher between 2016 and 2021, and we at UC are also very proud that he was once Associate Professor of Molecular and Cell Biology at the University of California, Berkeley. Welcome, Alan. Fourth judge is Jordan Felder, and Jordan is a senior also at Gateway High School in San Francisco. She will be attending UCLA in the fall, majoring in sociology. She is a part of many clubs and organizations, including the Boys and Girls Club of San Francisco, where she participated in the 2022 Youth of the Year program, representing her school's own clubhouse. After her undergraduate studies, she plans to attend law school to pursue her J.D. with a concentration in social justice. And our fifth judge is Robert B. Horowitz, who is the current chair of the UC Academic Senate. He is a professor in the Department of Communications at UC San Diego, where he has been on the faculty since 1982. That's a 40th anniversary this year. That's an amazing thing. He received his BA in Social Thought and Institutions from Stanford and a Ph.D. in Sociology from Brandeis University. So they are all here live with us at LinkedIn today. So thank you for all of our judges for being here, taking the time to join us. And I know that there's a very exciting program to come. And let me say, so a round of applause for all of our judges collectively. Thank you, judges. And then there's one final important judge, and that is all of you. Whether you are with us here in person today at LinkedIn or watching on our live stream, the audience will have an opportunity today to participate in Grad Slam. Once again, we will present a People's Choice Award, in addition to the winners selected by our panel of judges. So after the presentations have concluded, we will provide instructions on how everyone watching online can vote, and so you can keep track of your favorites and help us to choose the People's Choice Award at the end of the program today. So now, without further ado, I'm always wondering about further ado. You know, we're denied further ado so often, but we'll once again deny further ado, and we'll get on to Grad Slam. The research these graduate students are about to present to us represents years of hard work. From wherever you are, please give each of our contestants a warm welcome as they come up on stage. And I'd like to begin by welcoming our first contestant to the stage, Wei Gordon from UC San Francisco. Wei?
We eat too much sugar. I just wolfed down bowls of Cinnamon Toast Crunch to get myself extra jazzed for Grad Slam. But really, it's impossible to avoid sugar. Diabetes is the 8th leading cause of death in the US, and more than 1 in 3 adults are pre-diabetic. So how can we alleviate some negative consequences of a high sugar diet? We can learn something from mammals that specialize on a high sugar diet. These mammals include fruit-eating bats and primates. Though we know fruit eaters eat more sugar, yet lower their blood sugar faster than non-fruit eaters, we don't know how traits like this arose at the molecular level. Little changes or mutations in DNA can result to changes in traits. Traits are encoded by DNA sequences called genes. Mutations in genes change traits, but mutations in DNA sequences around genes called gene regulatory regions. Change traits too. You can think of these regions as conductors of an orchestra. If genes are the instruments in an orchestra that creates a living being, then gene regulatory regions are the conductors that control when, where, and how much an instrument is played. I'm not the first person to ask a fruit eater, "Hey, what sugar secrets are you hiding?" Previous investigators have focused on finding mutations in genes, but only a few have been found. Because no one has examined gene regulatory regions, the conductors, I look to find mutations there that lead to fruit eater traits. I analyzed DNA of over 100 mammals to identify sequences with mutations only in fruit eaters. I found thousands. The next step was to test if these mutated sequences are gene regulatory regions. I selected a handful of sequences and I put them in front of genes that can light up into cells. If a sequence is a gene regulatory region, then a light gene is instructed to produce light I can see. Every fruit eater sequence I tested gave light. I was lit with excitement. Did I find sugar secrets? To test if the regulatory regions I discovered affect sugar metabolism, I put the fruit eater sequences into a non-fruit eater, the mouse, and I'm examining mouse development and response to high sugar. My regulatory region database offers secrets for high sugar adaptation. This can assist therapeutic developments for metabolic diseases like diabetes. I'm not saying I found a reason for us to eat as much Cinnamon Toast Crunch as we'd like, but I may have found a reason life can be just a little bit sweeter. Thank you. Thank you. Thank you. Oh.
So we have a few. Turn my mic on. There we go. We have a few tough questions, okay? When you were first practicing your presentation, who was the first person you trusted as an audience, and what kind of feedback did you get?
Ooh, the first person I trusted as an audience. So my lab has actually a really strong history of students and postdocs competing in Graduate Slam and Postdoc Slam at UCSF. And I have to give a huge shout out to a graduate student who just graduated, Serena Tamura. She actually was a finalist in 2020, and then the competition never went through, so she never got to see it through. And so I reached out to her when I first was interested in doing this, and yeah, she helped me with a lot, and I'm forever grateful.
Fantastic.
And I know she's watching, so hi,
Thanks,
Serena.
Yes, fantastic. That's great. Mentors are so important, and I appreciate that. You don't want to play favorites, I'm going to guess, but what is your favorite fruit-eating mammal?
My favorite fruit-eating
You can
mammal.
whisper.
I mean, there's not a whole lot I can choose from, but I'm wearing bat earrings. I really I had some incredible opportunities in the lab I'm in with Nadava Hitub, who's in the audience today. But I will say I really like the dog-faced bats. They look like dogs, and they can actually climb trees too, like in a really disgusting manner, but it's really cool.
Great. So maybe another research project will be teaching you to fetch or something. But it sounds great. Sounds good.
That's a good idea.
Great. And tell me, what's your favorite thing? What's been your favorite thing about conducting research at your campus?
My favorite thing really is like the support that I've gotten from everyone around me. I feel really fortunate I do believe, and I say it all the time, that I've picked the best lab, and everyone in my lab is truly just pushing each other on, and I think it makes any research environment more exciting and more fun when you're able to feel like you can communicate to everyone around you. And I would say this is pretty true of like the UCSF community too. Like I like the way that the buildings are built, and it is, kind of forces you to interact with other people, but that's like how great collaborations happen. And so I think it's the environment that really fuels me.
It's interesting, so many questions we ask about how's this going or how does that work, and it turns out that it's the people. That's
Yeah.
really an amazing thing. Last question, if you were to be UC president, what project would you attack first? What would be the first thing you would do?
Oh my goodness. That's,
In fact, we can trade if you get
I
yeah.
mean, I really like your idea of talking about training bats to do fetch. So maybe I would fund that.
Let's go for it. Great to have a chance to meet you.
Oh.
Okay, great. Yeah,
Thank
congratulations.
you. Thank you.
Thanks. So now I know my homework for tonight is to get working on bat fetching. But before that, I want to welcome our second contestant to the stage. And this is Amanda Quirk from UC Santa Cruz. Amanda?
If we were to truly study a galaxy's life, we would need billions of years to watch it form and grow. Now, that is way longer than I want to be in graduate school. So, to figure out how galaxies change over time, astronomers like me have to act more like archaeologists. We look for clues that represent a tiny snapshot of how a galaxy has changed over time. Now, a whole lot can happen to a galaxy in billions of years. There are violent bursts of star formation, energetic deaths of massive stars, and collisions between galaxies. I'm particularly interested in those collisions called galaxy mergers or galactic cannibalism. Mergers tear apart smaller galaxies while feeding larger ones. They can completely change the way a galaxy looks and behaves, so they are a critical part of a galaxy's evolution. I look for signs of mergers in two nearby beautiful spiral galaxies, Andromeda and Triangulum. To study these galaxies, I observed stars in them using one of the largest telescopes on Earth. You see, the thing about stars is they're gossips. They give away many of their secrets just through their light. With cutting-edge technology, we can measure how fast a star is moving while we are millions of miles away. I leverage this to compare how fast young and old stars are moving. If there's a difference, we think that something must have happened in the galaxy between the time the young and the old stars were born, and that something just might be a merger. Using this technique, I found that the Andromeda galaxy likely had a major merger in the past 4 billion years. This is relatively recent, and actually changes what we know about the types of mergers spiral galaxies can survive. It also calls into question the interaction between Andromeda and Triangulum. So I've been observing Triangulum for the past 4 years now, and I've found that there's actually not much difference between how the young and the old stars are moving. This suggests that, so far, Triangulum has been left alone by Andromeda and by other galaxies that have passed by recently. But it actually won't stay that way forever. Triangulum will collide with Andromeda in billions of years. And it's not the only one. The Milky Way, our own galaxy, will also collide with Andromeda in the far future. We won't be around for that merger, but the work I'm doing today does give us a glimpse into the future of our very own galactic home. Thank you.
Congratulations.
Thank you.
You're on mute. Zoomer. Okay, there we go. I remember stargazing with my sons as we were growing up, and Andromeda was really special because you could see it with the naked eye.
Yes.
It looks like a smudge, but it's
Oh yeah, it's not that exciting
yeah.
without a giant telescope, but it's there.
But it's exciting that you can see something that big and that far away. So that was terrific. Nice to see it. Always nice to see it again. When you were growing up, what now are your favorite things to do when you're not conducting research?
My favorite thing is to snorkel. So I've been so lucky. The telescope that I use is in Hawaii. It's on Mauna Kea, which is a very sacred site to the indigenous Hawaiian community. So I'm very fortunate to be able to use it. And while I'm there, I try to also appreciate how beautiful Hawaii is. And so when I'm there for science, I snorkel. when I get a chance, and you can go at night and snorkel with manta rays, and that's one of the most amazing experiences that I've ever had.
Yes, I'm actually afraid for you, but good. I'm glad that sounds fine. So you mentioned manta rays at night that you've seen snorkeling. What other things have you seen that were really spectacular during that snorkeling?
There are sea turtles, lots of different kinds of fish. I'm not an expert. There are people in the crowd who know way more about fish, and I've been so excited to talk to them.
Great.
But it's just amazing. You know, I think astronomers, it's important for us to look back on Earth and see how beautiful and special our own planet is. So I
Yes.
try to do that.
It's great to have those two pastimes, one looking way, way out into the sky and in the future, or the past, excuse me, and then looking also down beneath the surface. That's really terrific. If you were going to tell me something that's really specific or that's exciting about conducting research at your campus, what would that be?
I think it is being able to work with other students. So there are a couple of programs at UC Santa Cruz that connect graduate students with undergrads and high schoolers. And whenever I'm stuck and thinking like, who cares? I'm just typing numbers on my computer all day. When I get the chance to interact with a student who is motivated, who tells me, wow, this is really cool, I want to know more, that is so rewarding to get me to continue to do my research. So I think that's my favorite part.
Fantastic. You're doing astronomy now. What did you want to be when you were growing up? What did you think about first?
Well, first I wanted to be a vet, but then I realized I faint when I see blood, so had to ditch that career idea. But I've always really liked math. I was fortunate enough to live in a place that had a planetarium and observatory, so I got to look at Saturn in a telescope, and that was really, really impactful on me. I think I was in 4th grade maybe when I first saw that, and from there, astronomy just stuck with me.
Fantastic. Sometimes you might have been looking in the sky and seen Regent Hernandez zoom by. That would have
I
been great.
should have been
So
looking.
who knows? That makes it a wave. And then you know there's a value of being able to explain research simply. Can you just describe that value? What's important about that?
Yeah, I think the most important part about science is communicating it with others. It doesn't matter how cool or innovative our discoveries are if we can't tell people about them so that they can learn and they can use it. And I'm really passionate about teaching, so I think teaching and communication have very similar skills. It's how do you translate a high-level concept into language that not only is understandable, but is interesting to people. And so I think it's really key, and I'm grateful to have had the opportunity to practice my teaching and communication skills.
Fantastic. Thank you very much. Congratulations. Thank you, Amanda. Our third contestant will now come to the stage, and our third contestant is Justin Lee from UC Berkeley. Justin?
Let's address the elephant in the room. It's 2022, and the COVID-19 public health nightmare continues to dictate how we live. Vaccines have helped prevent severe COVID-19 cases, but new variants continue to surface. At Berkeley, my research team and I are hard at work in the lab trying to develop a new treatment for COVID. Our ultimate goal is to help people and to stop this ongoing arms race with the virus. We first started our research by learning how the SARS-CoV-2 virus works. Shown here in red, it enters the body, hijacks our own machinery to become a copy machine, to produce tons of virus copies for more infection and spread. We then studied the genetic makeup of the SARS-CoV-2 virus, and we were able to find the key code in the viral RNA that allows the copy machine to run, and that all the variants, including Delta and Omicron, share the same key code. This was significant because now we had a target. With a target in our crossfire, we modified the existing genetic technology to make products designed to specifically disable that key code. I tested these modified products shown here in blue. They're known as locked nucleic acid antisense oligonucleotides, or just ASOs for short. Now, I was able to isolate just one of these ASOs that appears to accomplish our goal. The ASO that I developed can be inhaled through the nose. That delivers the treatment directly to the airways and lungs. Once there, the ASO binds to that viral key code, jams the copy machine, and stops the virus from making more copies. Next, while rocking the trendiest scientific safety gear, I tested the inhaled ASO in mice infected with COVID-19. First, on the top right, this is a lung sample from a mouse infected with COVID-19 that has not been treated. You can see the severe infection all over in the dark spots. Directly below is a lung sample from an infected mouse that was treated with the inhaled ASO. As you can see, the virus was almost undetectable in this lung sample compared to the untreated mouse. Look, we still have a lot of work to do. The research is currently under peer review. If it is published, then some scientists above my pay grade would have to apply the research in clinical trials to ensure it is safe and effective for humans. But can you imagine what a game changer it would be if we could fight this virus by just taking a breath? Here's to hoping.
Well, very good, congratulations, and all the
Thank you.
practice I've had jamming copy machines, now I know there's a real purpose for that. That's good. So what are your favorite things to do when you're not doing research?
Yeah, recently, I mean, ever since the pandemic started, you've really had to find your own, new hobbies, independent hobbies. I've really enjoyed picking up running. I think there's a real value in being able to just get outside, breathe in that fresh air, push out, all your negative energy, and just really just fill yourself with a lot of positive feedback. And then, a lot of time at home, finding new comfortable positions in bed or on the couch to stream Netflix. I've gotten really good at balancing my iPad on my stomach and one of these angles and tilting the head at the optimal viewing angle, and it's a great way to help fall asleep at night as well.
Great. Sound like an influencer. That's great. I appreciate hearing that. What did you want to be as a kid when you were growing up? What was your?
Yeah, it's funny. I actually, when I was really young, I wanted to be an astronomer, and there's so much expertise here, and great lunch conversation topics here. And doing this research a little bit, when we had to wear some of these big kind of suits to work with the virus, it was like kind of wearing an astronaut suit, but kind of not. We're still on Earth. My feet are on the ground. Then I realized I'm probably not the best with heights, so that may not be a great idea to send me blasting up far away from the ground. So one way to fulfill a dream while keeping my feet on the ground.
That's a great thing. I appreciate it. You tried snorkeling in that gear and you got it all tied together. Well, never mind. Maybe that's not
You
it.
gotta
Right.
look for tips and find places.
And then when you think about the ways that your research is going to impact the pandemic, what do you think is going to be the first thing that we're going to see from this?
Yeah, I mean, hopefully, as I mentioned, we need to have this published first, get it peer reviewed to make sure that it's also safe and effective in humans. But I think the big novelty to this research is that because we're developing an inhaled treatment, this really targets a population that may be a little needle anxious or needle averse, and gives you a very direct treatment for a virus that causes disease in your respiratory tract. So we're really kind of narrowing down where we're targeting and hitting the target where they would have the most effect. And so I think that's where, hopefully down the road, that's where we're going to have the most impact in our communities and around the world.
Great. And you've been working with your graduates, your program leaders very actively. What's something that your graduate advisor doesn't know about you?
Oh gosh.
Well, we're being,
yeah,
okay, yeah, right. Okay.
I definitely make my own hours. So if you expect me to be there at 8:30, I am not there at 8:30. Depending on when the caffeine kicks in, it could be 10 a.m. Depending on when I get out of bed, it could be noon. But yeah, I make my own hours and work that way. And hopefully he doesn't come knocking on my door when I'm not there.
I'm sure what the advisor sees is the quality of your work, which is obviously excellent. Congratulations. Great to meet you.
Thank you.
Thank you, Justin. I'd now like to welcome our fourth contestant to the stage, Meghana Pagadala from UCSF.
Omicron, Lambda, Delta. With COVID, we've heard the word variant a million times, but what does it actually mean? A variant is a sequence change in a gene that makes it genetically different from the original sequence. Now, with these COVID scariants, we've seen how these small sequence changes can make a big impact on the pandemic. Now, you and I are variants on a similar level. We all share the same human genetic sequence, but when we were born, we inherited these sequence changes, our germline variants, from our parents that make us different from one another. For example, I have germline variants that make me 5'7", whereas you might have germline variants that make you 6 foot. You might have germline variants that make cilantro taste like soap, whereas I love cilantro. My research has shown that these germline variants can change the way that we think about cancer. Cancer, like COVID, is an invisible enemy. Unlike COVID, though, it's not foreign, but rather arises from within your own normal cells like an insurgency. Right now, our best weapons against cancer are chemotherapy, surgery, and radiation, which are unreliable, expensive, and harmful. Instead of throwing these destructive grenades against cancer, wouldn't it be great if we could catch it before it turns into this full-fledged army? Do you know what your first line of defense against cancer is? It's within you, your immune system. Your immune system is like a highway patrol protecting you from these cancer insurgents that are trying to take over and invade your city. Unfortunately, in most individuals, cancer is really good at hiding from your immune system. But in some lucky individuals on the left, their immune system can catch these cancer insurgents, bring them out into the light, and eliminate them before they can ever form an army. For my research, I looked at germline variants for individuals in both of these categories and found that they were related to a special immune task force known as antigen presentation. Antigen presentation is like a highway checkpoint. Officers are checking every car for a valid driver's license. If something seems fishy, the sirens go off and other officers are called to the scene. This is one of the most important immune processes protecting us from cancer. And I found germline variants related to antigen presentation could predict melanoma and prostate cancer risk in over 500,000 individuals. So what does this mean? By looking at your sequence changes, we can see if you're going to get melanoma or prostate cancer earlier, which means less chemotherapy and saving more lives. Now, this is just one finding from my comprehensive germline analysis. But with my research and the power of 23andMe, we can catch cancer earlier with just a spit in a cup or a nasal swab. I mean, with COVID, aren't we already kind of used to it? Thank you. Thank you. Thank you, Dr. Drake.
So a few things, Meghana, nice to meet you, and great research. Just start with a fun fact. If you could meet any person, dead or alive, who would that be?
Oh, if I could meet anyone. Oh, that's a really hard question. There's so many people that I would want to meet. I think this is a hard one. I think I would meet, right now I would meet Elon Musk and ask what he's going to do with Twitter after acquiring it. That's what I would do.
I think he works here or something. No, maybe not. Maybe
We
I
should
got
just go, like, look
my
for that.
tech companies confused. Great. So we'll let Elon Musk know that,
Yeah.
and then we'll find out. I'll have to whisper it to you. That's great. And if you're thinking about the news lately, has there been a particular research study or finding that has fascinated you over the last bit of time?
Oh, I think there's lots of cool research coming out, especially in cancer. I think recently what's been interesting for me is spatial transcriptomics. And so that's where you can actually visualize the tumor and also be able to molecularly profile it. And I think that that will be really fun to see where that technology goes in the future.
Fascinating. Yeah, absolutely. And when you were growing up, what did you want to be?
When I was growing up, I actually wanted to be a dancer. And so I still continue to do dance to date now as relaxation from research. So
that's a good thing to do. And if you were thinking of students, we have some students who are about to enter their undergraduate years. If you were going to talk to students about whether or not they should pursue graduate school, what kind of things would you tell them about that experience that have been the most enticing and supportive?
Oh, that's a really good question. Especially because I myself wasn't sure if I was going to go to graduate school until later in undergrad. But I think one of my favorite parts about graduate school is it really gives you the opportunity to develop individually. I feel like I came into graduate school and I was not as all confident in public speaking or in other aspects in my life. And graduate school really helps you to hone in on your skills, be an independent thinker, and become confident at the end of that. And so I think it's really fun in terms of that. It is very long, but I think if you can get paid to go to school, I think that's really cool. So.
That's a great thing. And you said you were going to be you were interested in being a dancer, and now you're a scientist. Was there a time when you made a decision to pursue one track or the other?
Yeah, so it started actually very early for me. So when I was in middle school and high school, I actually grew up on the border of Iowa and Illinois and went to a very small private school in Iowa. And so science fairs were really big in my middle school and high school, and I would go to the state science fairs. And I feel like doing that and presenting my research was such a great experience. And then I got to have my first academic research experience at St. Louis University School of Medicine and actually met an MD PhD there. And that really piqued my interest to be like, again, if I could get paid to go to school, what a great idea. So I thought that was.
And you said you were in the Iowa State Science Fair. You actually won, if I'm correct. So
Yeah.
what was your, so that was a good thing. You're modest about that. What was your winning research?
So my favorite research, or the one that I won for in 6th grade, was the blade shape of a wind turbine and which one was the most energy efficient. And I still think today that is my biggest contribution to science because it is the best experiment I put together. The blade shape, I made sure that it was the same surface area, and so it was very controlled. I repeated it several times, and so that was what my topic was on.
A wonderful topic, and we have a lot to learn from that.
Yeah.
Congratulations, great to see you.
Thank you so much.
And now I'd like to welcome our 5th contestant to the stage. Our 5th contestant is Alice, excuse me, Alice Dien from UC Davis. Alice?
Who else was shocked by the price of their energy bill in January? At first, I thought I had left the oven on for a week or something. But then I found out that natural gas prices have doubled since last year. Great. First it was the gasoline, now it's the natural gas. Do you know what's next? Food. My name is Alice Dien, and I study how we use energy to make food. Specifically, I specialize in one of the most energy-intensive processes, drying. If you had a cup of coffee this morning, or if you've ever eaten almonds, or walnuts, or even just rice, you can thank drying. Without it, all these products would have spoiled within 1 week. In fact, 60% of the calories lost globally today happen because of things that were incorrectly dried. But how does this all relate back to energy, right? I asked the rice processor in Sacramento how much she spent to dry the rice in her facility last year. That means bringing it down from 20 to 13% moisture. Her answer, nearly $500,000. Now imagine what that's going to look like when it doubles. Almost 1/4 of the entire US food industry's energy consumption comes from drying. And not only does drying use a lot of energy, but it also releases greenhouse gases from burning gas to produce heat. Heating our dryers is also heating our planet. So what can we do about it? Who has ever been tempted to eat one of these delicious looking things? That is a silica gel package, one of many examples of a desiccant material. Desiccants can remove moisture from their surroundings, and when used with a fan, create a flux of dry air. In my research, I'm designing a new drying system that uses desiccants instead of heat. I took a variety of crops, including corn and rice, and tested desiccant effectiveness using a dryer we built in our lab. My results show that not only do desiccants work, but with them, we can replace burning natural gas with electricity and make drying cleaner and cheaper. Now, I can plug my system into solar panels and operate completely off-grid. Imagine what this would mean for farmers that don't have access to the electric grid yet, but that need to dry their staple crops. Desiccant drying would be a game-changing solution for three of the biggest challenges humanity is facing: food safety, food insecurity, and climate change. Thank you.
Appreciate that very much, Alice. Great to see it. And I've actually never been tempted to eat one of those packages, just so you know, they don't look somehow tasty to me. When you were growing up, what did you want to be?
I actually wanted to be a musician. I'm a saxophone player, so I was doing like school and music for a very long time. And when I was 17, right at the time where I had to decide, am I going to go to college, or am I going to pursue music? I just decided that I couldn't leave science out of my life, so I ended up going to, I grew up in Spain, so it's a different setting than here. I know there's music here in college.
Yeah, there's music here too, but that's
Which is very nice, actually.
It's a good thing. If you, we've all had this quarantine we've been going through the last couple of years. Did you pick up any interesting hobbies during that time?
Yeah, I started growing plants, which still blows my mind. I was not able to keep a plant alive before the pandemic. But now I have my living rooms full of plants. I started my little veggie garden in the backyard, and they're doing well.
Fantastic, fantastic. And speaking of conducting research, was there anything particularly surprised you during your study? You got interested in a very, very interesting project. What things did you learn along the way that were surprising?
Yes, so it's an interesting topic in many ways. There's applications for desiccants already, and no one's been like developing these sort of like dryers. So that was very interesting to me. But I read some articles on using desiccants as a way to capture water and then like access that water and use it in places where there's a drought or something. So I thought that was very, very interesting. I was also reading like desiccants remove moisture from the ambient. So there's places that are very dry, like California. You can use desiccants to like remove that water from the ambient, even if the relative humidity is very, very low. So there's a lot of like very interesting applications for the future of desiccants. I'm excited about that.
Very interested in the future of desiccants. I was actually thinking with your talk that maybe if somebody produced an edible desiccant, well, never mind. I won't. I'm just trying to go forward. Let's say that you woke up one morning and you were magically the head of the White House Office of Science and Technology. Okay, what's the first thing that you would think about doing then?
Well, I guess I would wonder, why did they put a French person here? But yeah, I think there's a lot of issues around like food security. I became interested in like, I do a lot of international work. And then I found out that Davis, where I live, there's like between 30 and 40% of food insecurity just in that little town. And I think that's a very important topic to address, because at the end of the day, we all need to eat to survive. So yeah, that would be one of my first things to think about and address, like what are programs, technologies, policies we can do for it, and also for the farmers who are really struggling with climate change. Especially.
Well, you'd be a great director of the Office of Science and Technology. Thank you. Congratulations.
Thank you so much.
I'd now like to welcome our sixth contestant to the stage, and this is Rachel Sousa from UC Irvine. Rachel.
I remember remember sitting in math class during high school and hearing students ask, why do we have to learn math? When will we ever use it? Math has always been my favorite subject, but I will admit that I too questioned how we could use math outside of a classroom. It wasn't until I pursued a degree in mathematics that I fully understood how we could apply math to solve real-world problems. Now, I use math as a tool to understand biological systems. As a mathematical biologist, I aim to build a bridge to connect the seemingly separate worlds of math and biology. I specifically focus on cancer biology, as cancer is one of the leading causes of death worldwide. One promising cancer treatment has been to stimulate a patient's own immune system to better fight their cancer. This is done with drugs called immunotherapies. There are many immunotherapies available, and new ones are constantly being developed. However, certain immunotherapies only work in half of patients, and some patients have bad reactions to immunotherapies. We don't know exactly why this occurs. The human body is a complex system with many different interacting parts that are hard to identify with biological experiments alone. And that's where math comes in. Math provides an ideal framework for studying cancer by modeling the interacting components in ways that allow us to control and predict different aspects of the system. In my research, I am developing a set of equations, or a mathematical model, that will allow us to explore the complex cancer immune system interactions of the human body and enable us to predict the most effective immunotherapy combinations. Using my model, I can perform virtual experiments where I can study and influence the dynamics and interactions between immune cells and cancer cells. My goal is to use math to identify the key features that distinguish the ability of the immune system to eliminate cancer, and those features that allow cancer to evade the immune system and persist in the body. By understanding these differences, I will be able to predict patient responses to immunotherapies and develop new combinations of drugs, doses, and schedules that can optimize treatment response and minimize unwanted side effects. Then, I can work with an experimentalist to test my model predictions in mice. If my model predictions don't match experimental results, then we can fine-tune the model using this new data. On the other hand, when my model predictions match experimental results, then we can use my model to inform human patient treatment. Unlike cancer, math is predictable, and we can use the power of mathematics to better understand the complex biology of the human body. By sharing my research, I hope that if you were one of those students questioning why we have to learn math, you can now begin to see that we can use math in all sorts of ways, including to revolutionize cancer treatment and save millions of lives. Thank you.
Great, thank you very much. Great to see you. When you were growing up, what did you want to be? What were your career directions?
I wanted to be an actress. So I did a lot of plays and musicals growing up, but I soon realized that I couldn't sing and I really wasn't that good of an actress. So quickly went another path.
Great. What age about was that? When did you make that?
I think the last play I did was in high school, and I originally didn't get the part, but then somebody dropped out, so I managed to get a part. So I was like, okay, maybe this should be it.
Okay, great. That's fantastic going forward. You work very closely with the people in your lab and your research advisors. I asked this to one of our other winners. What's something that your project director doesn't know about you?
That's a good question. I learned this about myself last year when I was in a developmental systems biology class. I have what's called the brachydactyly type D, which I believe is a mutation in my HOXD13 gene that gives me wide thumbs. So I have wide thumbs that are very short and stubby as well. And so I feel like people don't know that about me unless they really look at my hands and ask me about it.
Well, congratulations on that. I'm learning something. That's learning something new. And you've been through your program. You were going to be an actress. You focused on math and science. Great. As you're moving forward, were there times that you had doubts about the math and science career, and how did you get by those?
I don't know if I necessarily had doubts. I originally wanted to be a high school math teacher, and so I went into undergrad pursuing math and education. But then shortly after that, I discovered the field of mathematical biology. And I was just fascinated by it. And since then, I've just kind of been following my nose and started doing research. And then I wanted to keep doing research, so I went into grad school, and now here I am, and it's just been working out so far. So far, so
great.
good.
Well, and if you think about the experience that you've had in your graduate program, what would you focus in on as the most compelling?
I think I have to reiterate what some of the other contestants have said. It's just that the people there really make the program. UCI in general is just a great campus, but then the program that I'm in, as well as the 2 labs that I'm in, just have such great people that are so caring and kind and so collaborative too, that really foster such a nice community that it really makes it worth it to be there.
Fantastic. And I had this question was given to me. It said, "What Hogwarts house do you belong to?"
I've never seen all of the Harry Potter movies, so probably not a good question to ask.
You know what's great is you've seen more than I have, so I would have no idea which Hogwarts house to look at. Which of you follow? You mentioned being interested in theater and arts. Are there some plays that you've seen that were really moving or meaningful to you?
Oh, I wasn't good about going and seeing other plays. I think the only real play I've seen was I saw the Spider-Man play in New York, and I think 8th grade, and it was really cool and really fun. So again, this is probably why I shouldn't, like, theater was not a good direction for me.
Right. But let me say, it's not. There are a lot of good plays and things out there, so there's great things to do there.
Yeah.
And we're very excited about your research. Will you give a shout out to the University of California when you're able to cure cancer?
Oh, absolutely.
Fantastic. Congratulations.
Great. Thank you so much.
And now it's time to meet our 7th contestant. And so I'd like to welcome Jessica Wilson from UC Merced. Jessica.
Place your right hand over your heart and feel your heartbeat. Could you tell your heart is working harder than it should? The unfortunate reality in the United States is that more than half our population is estimated to have high blood pressure, but for too many, a diagnosis comes too late. We've learned over the years that high blood pressure is not caused by one thing, but a combination of issues: obesity, smoking, diabetes, the list goes on. And because there are so many conditions that can cause high blood pressure, finding a cure is very difficult. We've all heard that diet and exercise are great for heart health, but what if you already have high blood pressure? Losing weight helps, but it's not enough. So high blood pressure is mainly controlled with meds, lots of meds. One lowers cholesterol, another removes excess water, one med, one action. This means a lot of different meds are needed to control high blood pressure. And do you know what's scary? The latest drug therapy was developed over 30 years ago. But recent legislation has finally allowed researchers to explore the potential of a plant you may have heard of. This is a cannabis flower. 7 independent studies found that people who admitted to smoking marijuana often had a lower body mass. You may be thinking, how? Isn't smoking bad for you? Yes, but cannabis contains over 400 different chemicals, the most abundant of which is cannabidiol or CBD, which doesn't get you high, but seems to do a lot. CBD can reduce pain, inflammation, and seizures. A handful of studies focused on CBD's effects on the brain reported that the treatment lowered blood pressure, blood cholesterol, and body mass. At UC Merced, I study high blood pressure with Dr. Rudy Ortiz using a rat model that mimics the human condition. I wondered if CBD could have the same effect when blood pressure is already high. So I fed fat, diabetic rats with high blood pressure CBD for a month, and it brought their blood pressure down. Now here's the exciting part. CBD also reduced their belly fat, lowered their cholesterol, and made diabetic rats more sensitive to their own insulin. We think that CBD does this by activating the body to consume its own fat stores, which would make CBD an incredible weight loss supplement at the very least. What I'm most excited about, though, is that CBD didn't do just one thing here. It improved multiple causes of high blood pressure. One med, many actions, many benefits. To be clear, I'm not saying CBD is a cure for high blood pressure, but this research is helping to address the urgent need for new therapies before it's too late.
I went on a campus visit to UC Merced and I saw rats lined up to get into a lab. I think it was yours. When did you
We're very
first,
popular these days.
when did you first become interested in this particular line of research?
To be honest with you, I'm a California native, so I've watched the debate over cannabis and legalization of cannabis and all of the arguments that have passed back and forth. And what I noticed, especially my advisor as well, of course, was that we don't really know what it does. And that's really interesting. And moreover, because the access to cannabis has become so pervasive, it's easily accessible now. And as you know, there's a recent federal decriminalization verdict that happened earlier last month. I know more and more people in my immediate proximity who are consuming products for all kinds of things that they don't really know if it works. So, I mean, this research is really helping to bridge that gap and help us understand what exactly cannabis does.
Fantastic. What did you want to be when you were growing up, when you were thinking about going to school and all that? What were your directions?
I really wanted to be a veterinarian.
There we go.
And I worked as a tech for many years, and I realized that it was the bench work and the asking and answering of questions that really intrigued me, not necessarily the day-to-day practice. So this gives me an opportunity to consistently learn by pursuing grad school.
Fantastic. Let's say you could have a superpower. What superpower would you have?
Brilliant memory.
Okay, fantastic. A brilliant memory was a superpower, just so you could, it's a good thing. I was going to ask another question, but because
it's gone.
And what do you plan to do after you graduate? That's coming up in a bit. So what are you thinking of doing in the next phase of your career?
Probably keep normal business hours. That will be a start. But certainly my primary goal is to move closer to my family. I'm a native of Southern California, so where I'm at is a bit far. And certainly the pandemic has really helped me to adjust my priorities. Not spending Christmas with my family was very difficult. So moving closer to them is really a priority in my life now. So see if we finish.
We'll see, we'll finish
We're
soon.
trying to finish on time here.
We'll finish on time. And then maybe my last, if you were thinking, if you could think of advice that you'd received during your career, what was some of the best advice you've received?
It's not necessarily about pursuing a project that you hold so close to your heart. Follow the questions. That's what I started to get lost in the sauce early on in my graduate career and focused on, I want a project on this specifically. And I realized that that's not really what I was interested in. It was the pursuit of knowledge. That's what I'm interested in. So yeah, don't get lost in the sauce on the specific project. Ask questions.
I think keeping our eyes and our minds and our hearts open is always good advice moving forward. Great to meet you. Congratulations. Thank you. And now we're on to our 8th contestant, and I'd like to welcome Claire Whitaker from UC Riverside. Claire.
I love fresh produce and I hate food waste. When you hear food waste, what comes to mind? Leftovers in the trash? Produce gone bad? A landfill? Or I should say landfills, because around 1/3 of the world's food goes to waste every year. That's 1.3 billion pounds. So it's the worst feeling when I reach for a strawberry and see that it's moldy. It's not just strawberries either. Grapes, tomatoes, they all seem to get the same fuzzy gray mold. That gray mold is caused by a fungus called Botrytis cinerea, and it can grow on over 200 types of plants. Part of why it's so common on produce is because there are no management strategies that can fully remove the fungus once it infects a plant. We need new tools that target the systems the botrytis fungus uses to infect and spoil produce. See, botrytis produces proteins designed to interfere with the plant's major defense mechanism, their cell wall. These proteins work to tear apart the plant cell wall, eventually breaking the cells open and killing them. But it seems like there are other fungal proteins that aren't following this path. They're breaking down plant defenses from inside plant cells. I'm working right now to explore the possibility that botrytis is sending proteins into plant cells using a system like Amazon bubble mailers. The proteins are packaged up inside fungal cells. Then they're released into the space between cells. The plant cells see these little bubble mailers and accept them, probably with as much excitement as one of us when we get a delivery we forgot we ordered. Unfortunately for the plant cell, this is more like getting sent an envelope full of glitter. It makes a huge mess that the cell then has to spend forever cleaning up. My research is working to determine exactly how this system works. Once we know how the fungal bubble mailers are stamped, shipped, and delivered, we can develop new tools to disrupt the system. These new tools may take several forms, from sprays to prevent fungal growth from starting, to something like soap to rid your berries of moldy spots. We could even teach plant cells not to accept the bubble mailers using genetic engineering. Now, this won't just save all of you from moldy produce, it will also prevent produce from molding before it even gets to you. Remember that around 1/3 of the world's food goes to waste every year? 1.3 billion tons? While some of that loss is due to other causes, like insect pests, a significant portion is due to fungal infections. My research will mean that less food will go to waste globally. Thank you.
Appreciate your research and the pictures. If you're at the grocery store selecting strawberries, how do you pick them? What do you look for?
What I do is I like making overnight oats for breakfast. So if I'm buying strawberries, I'm buying them specifically to put them in my overnight oats in the morning. So I buy the ripest, reddest, freshest strawberries I can find. I take them home, I cut them up immediately, and then I freeze them. So I can heat them up in the morning and then add them to my oats.
Okay, I'll write that down and let you know how it works. That sounds great. That sounds great. When did you decide on pursuing science as a career or as a method of study?
I never really made a conscious decision. My father is a chemistry professor. He teaches organic chemistry. So as a small child, I grew up playing with molecular models, the little atoms that they give you in organic chemistry, the little kits. I grew up playing with those. So it was kind of always something that I knew I wanted to do. But then, as someone mentioned previously, the science fair, that was a big thing for me and my dad to bond over in middle school. So I did the science fair, and then by the time I was applying to colleges, I knew that I wanted to go into some sort of science career.
And what was your favorite project for science fair?
I really liked the one, I was looking at hydrophobicity of polymers. So I grew up in Annapolis, Maryland, which is on the water. So I was looking at different polymers that had different characteristics to see if they could be useful in plugging holes in boats. So it was essentially a piece of wood with holes drilled in it and a bunch of different stuff shoved into each hole to see if it would absorb water while not. It was balancing to figure out which one would be the best in a boat.
Fantastic. I'm trying to put everything together. So if we had a fruit that was a dessicant, no, never mind. I
would eat it.
I was trying trying to tie them all together. I asked one of our winter colleagues what they would do if they woke up and were the head of the Office of Science. What about if you were the president of the National Academy of Sciences? What would you think? How would you direct the science profile of the country forward? What do you think we should be looking into?
I think that one thing that I would do is try and work on the diversity and inclusion efforts that the National Academy is already working on. I think that science loses a lot when we don't have scientists from diverse backgrounds. So actually having the National Academy make an effort at the lower levels, like encouraging high schoolers to go to college and college students to go to grad school. I think that if the National Academy could like step outside of the academia bubble for a second and work on pulling in all sorts of different people into the academia bubble, it would make for an overall better overall situation.
I think that's terrific. I'll make sure they're listening. Take care. Congratulations. Now it's time to welcome our 9th contestant to the stage. This is Geoff Meyerhof from UC Santa Barbara. Geoff?
I have a friend who stays up way too late each night, watching Netflix and eating snacks. This same friend cannot for the life of him make it to his morning meetings on time, no matter what he tries. We'll call this person friend A. But on the flip side, my other friend, friend B, is just the opposite. This guy effortlessly falls asleep at 9:30 every night to make it to his oppressively early 8 a.m. class with minutes to spare. Now, the reason these two friends are different might have nothing to do with their respective levels of discipline or effort. Instead, it could be that they have different circadian rhythms. Circadian rhythms are the natural 24-hour cycles our body produces to align our sleep, eating, and alertness with the rising and setting of the sun. And unfortunately for night owls, most of society operates on an early bird schedule. In fact, there's a growing body of research showing that people with night owl tendencies tend to have lower grades in school, and that night shift workers have much higher rates of diabetes and Alzheimer's disease. What I study are the genes that regulate differences in circadian rhythms. And in particular, I study genes in the eye that affect these rhythms. So the eye does two things. The first is vision, which comes from the rods and cones. And the second is to reset the clocks in our brain each day to light. Now, these clocks in our brain are the reason that we can often wake up at the same time each morning, even without setting an alarm. And they're the reason that when we fly across the country to a new time zone, we get jet lagged. So it turns out the way the rods and cones sense light and the way the ganglion cells sense light is totally different. In fact, compared to the rods and cones, ganglion cells sense light in a way that's much more similar to how the fruit fly eye senses light. So to study how the eye regulates circadian rhythms, I use the fruit fly. I and my lab mate, Monglin Lee, have identified a fly eye protein that when mutated, extends its circadian rhythm from 24 to 28 hours, and as a result, transforms it into a night owl. As an example of this transformation, here I'm showing their daily feeding. On top in blue is a normal fly. And you can see that it does most of its feeding right before the transition from daytime to nighttime. And on bottom in red is our eye protein mutant. And unlike the normal fly, it feeds well into the night. Whether it also likes Netflix is unclear. But at least the night owl has someone to share dinner with. With these insights from the fly, we hope to one day be able to apply our findings to humans. The ganglion cells in the human eye have a protein that's very similar to the one we're studying here in the fly. By controlling this protein, we may one day be able to reset our circadian clocks at will, eliminating the ill effects of shift work and allowing us to keep whatever schedule we please. Thank you. Amazing.
We're just adjusting. Hold on a moment. I'm adjusting. So Geoff, a couple of things. First, congratulations on the great work. What's your favorite thing about doing research at UC Santa Barbara?
Oh man, So I think my favorite thing about research is, the opportunity to kind of come into work every day and try to be creative. I think that's super fun. And my favorite thing about, doing research in Santa Barbara is the location, right? I mean, it's totally beautiful, right by the beach. I can look out on the ocean, from my lab. And then, the last thing, as everyone has said, is it's the people that make, all the difference. I've got, amazing lab mates and an awesome mentor. So it's been, yeah, a lot of fun.
And what were you interested in doing when you grew up? When did you think about this pathway?
Yeah, actually, I had no idea what I wanted to do when I grew up. It really, wasn't until, maybe late in high school that I thought, okay, biology might be something that is interesting. And it's, totally serendipitous just because I had some awesome mentors at the time.
Great thing. And then if you're thinking, you've done this research on circadian rhythms, quite interesting. I was overseas a couple of days ago. I'm still not sure what time it is.
Yep.
So it's great. What were some of the things that you learned that you didn't expect as you were doing the research?
Yeah, I mean, I think one really interesting thing about circadian rhythms is, just how connected they are to health. So, if you mutate some of the genes that give animals these intrinsic circadian rhythms, they're a lot shorter lived and a lot less healthy. And alternatively, if you try to do things that improve their circadian rhythms, they get a lot healthier. So I think that was really surprising to learn about.
That was great. If you were going to meet anyone who's alive now or has lived, who would that be?
Oh man, let me think. Okay, this is a pretty niche answer, but there's this one like totally prolific biologist named Seymour Benzer who is like the first person to identify a gene that controls circadian rhythms. So I think like if I could meet him, that would be awesome to pick his brain.
I was hoping you were going to say Elon Musk, because we could just
This
just simplify things. But that's okay. It sounds like somebody we could call up. So what do you think of doing in the next phase of your career?
Oh man. Okay. Yeah. I guess the next step is probably going to be, a postdoc, hopefully in San Diego, so I can go live with my girlfriend. Yeah, I think that's the game plan.
That sounds terrific. And during this time you've been doing this work, it's fascinating work. You mentioned people getting up and going to bed at different times. People suffer from insomnia. Is that something you've ever suffered from? Is there anything that you do to help with that?
So I'm super lucky. I am a great sleeper. Yeah, but I think that, there are lots of tips and tricks for people with insomnia. I think I'm not really an expert on human sleep, but I think what the experts might say are things like, don't stay on your phone when you're in bed, and just try to make the bed a place where you're focused on falling asleep, and don't eat before you go to bed, and all the kind of grandmotherly advice.
And grandmothers always have it right, don't they?
Yeah.
Great to meet you. Congratulations.
Thank you.
And finally, I'd like to welcome our 10th contestant to the stage. This is Kaiser Rutledge from UCLA.
Smell is a potent wizard that can transport you across thousands of miles in all the years you have lived. Helen Keller. We can all relate to the power of smell, the power to set us at ease, or as Keller said, transport us back to a moment in time. For me, it's the smell of my Nana's house, or rain in the mountains of Asheville, North Carolina. While we can all recognize smell's influence, as humans, smell isn't our primary sense for how we navigate the world around us. But for fish, it is. Imagine being this skate, a relative of a stingray, in the deep ocean, using your sense of smell to find food, mates, and even warn you of incoming danger. This is the world these fish live in, and they have evolved one of the most acute noses in the animal kingdom. Just how sensitive are they? Up to 100,000 times more than us. That's better than the best canine detection dog. As a group, they also have the most diversity in nose shape and size of any other group of fish. Here's an up-close image of their nostrils. See those two little holes and flap-like structures?
And while we know that this fish has an excellent sniffer, we don't know how they actually smell, the mechanism. You see, a fish's nose isn't connected to their throat. It isn't involved in breathing, and there's no pump for bringing odor into the nose. On top of that, we don't know why they have so many different types of noses. My PhD research at UCLA aimed to explore exactly that. Why are their noses so weird? And why do we care? Well, this isn't only interesting from an evolutionary biologist's point of view, but from a design standpoint. An efficient odor sensing device that doesn't require the power of a pump, this could be a great model for artificial sniffers in water or air. Imagine having the power to sniff out military threats, pollution, and even check for air and water quality. To answer these questions, I worked with biologists, engineers, museum staff, and even researchers with the US Navy. I then created anatomically accurate models of stingray heads by 3D printing CT scans. Then, using high-powered lasers to illuminate all these tiny particles in water, we can visually see the fluid dynamics of odor capture and watch those green particles go into the nose. And what do we find? That not all noses are created equal. Some are better than others, more dynamic. This one, the weird flap, helps this animal capture odor faster. These designs are simple, innovative, and can be easily incorporated into new technology by simply mirroring this shape. The results of this research is already shaping the designs of new underwater technology at the Navy. Think underwater vehicles that can detect harmful chemicals in water. So, the next time you're transported back in time by your favorite smell, I hope you remember stingrays, and more importantly, how we can look to nature for innovative solutions. Thank you.
And Kelsi, I appreciate your coming up. I said your name wrong.
Oh, that's fine, no
Kelsi, great.
worries.
John Travolta for a moment. Here we are. Thank you. Congratulations. So
you.
a couple of things. First, fascinating work. And there was a new species of ray that you discovered. How did that feel?
Yeah, that was great. So during my master's, I was studying the general morphology. So I came in as a marine biologist, kind of studying the general shape and form of function of these fishes. And then I got into engineering and how they work a bit later. But I found myself, staring very intensely at these creatures, why they look the way they are. And their noses are so different. They're so drastically different from one species to another. And as a biologist, we also classify species based on their noses. Show me any ray or skate and I'll be able to identify it just based on its nose. So what's going on here? We don't know how the nose works. We don't know why it looks so different. What are the correlations? And so I was looking at these noses and so I ended up describing a new species of ray that has a different nose and snout. And so I got to name it, and I named it after my advisor who supported me. But then that really led me down this path of collaborating with other disciplines and learning a lot of engineering to understand how this works, and then ultimately how we can use that knowledge to advance technology.
Well, congratulations on that. So a round of applause for discovering the state nose. I remember reading something about people at the zoo, and they had a kind of a monkey island with lots of different monkeys on it, and had the people there, and they said the people think the monkeys all look the same, but actually the monkeys are vastly more different than the people looking at them are if you actually study them. So it's a fascinating thing, and great that you were able to do some of that work. How did you pick UC for your work?
Yeah, so I'm originally from North Carolina, as I mentioned, and I just kind of applied broadly across the country. And my mentor, he does a lot of work with fish, and I knew I wanted to do something with fish because they're the most diverse vertebrate. There's the most number of species, so there's lots of adaptions there. And so he did stuff with stingrays and rays. I was like, this is a cool group. So I ultimately ended up choosing that based on him.
Fantastic. When you were growing up, what were your interests when you were an adult? What did you want to be when you grew up?
Yeah, so I wanted to be a marine biologist. So I grew up in the mountains, but the coast was always just such a place of wonder for me. So I've always kind of been into the ocean.
Great. And you mentioned a great relationship with your graduate supervisor. What's something your graduate supervisor doesn't know about you?
Possibly that when I originally moved from North Carolina to California, I had a pet turtle, and I smuggled him on the plane here. So my turtle made it across security. but I don't think he knows that.
I'm sorry, the ground rules for things you share. No, never mind. Let me, I hope that you made it and I hope that Turtle made it too.
Yes.
Great to see you and
Thank
congratulations.
you. Thanks so much.
And now I think that is the end of that first round. We have a chance, having heard from Kelsi, to hear, after having your certificate, to hear a little bit about yourself. So I think we're going to have something up for that now, if I'm correct. Oh, I keep going. Now tell us a little bit about yourself. Well, that was the prior page. Thank you, contestants. Okay, great. Here we are. Good. There's a new page. Thank you again. Thank you again to all of our contestants. Let's pause for a moment, look at our contestants, and say thank you, contestants. You know, we say contestants, but we know these are all winners. So really, congratulations for winning your campus competition, and really for the great presentations today, and actually the great work and the great career trajectories that you've all really showed us today. So really terrific work. I was told at the very beginning, I know I mentioned to the group that we had another very special judge who'd be weighing in on the proceeding. So now it's time for everyone who's weighing in to get in on the action and vote for which Grad Slam presentation you liked most. It's time to vote for the People's Choice Awards. We've now opened the online voting, so do not hesitate, hop online and vote. The web access for online voting is displayed on your screen. So please go to ucal.gs/vote. And again, that's ucal.us//GS vote. Voting will only be open for 5 minutes, so vote now. Get to it, and the top vote getter will be awarded $1,000. So that's terrific. In that way, this person will have a wonderful People's Choice Award. So I think that we are setting up the interview area, and I see that the interview area is largely set up. A few facts of my own to share, which I'll do now during the voting, if I'm correct. I see a furrowed brow. Great. Okay, then great. I appreciate that. You know, when in the one thing in the pandemic time when people are wearing face masks, they can't mouth words to me or wink. You know, so I appreciate the okay. So let me say, Grad Slam, I see we have our interviewer. Grad Slam is all about innovation and research. And here's some interesting innovations and breakthroughs that came out of UC research over the past few years. Cochlear implants, that's for people who had lost their hearing. Blue LEDs, an incredible invention, incredible discovery that really will change the world moving forward, changes energy use amazingly. The wetsuit, of all things, that's great. The discovery of vitamin E. UC performs nearly 1/10 of the nation's academic research on an annual basis. Scientists from UC San Diego have recently developed the world's first biodegradable shoe. It's just that we're going to have edible desiccants soon. Now we have a biodegradable shoe. It was a postdoc at UC Santa Cruz and a UC Merced alumnus, Brandy McCune, who found that California could save 63 billion gallons of water a year, enough to irrigate 50,000 acres of farmland or meet the residential water needs of more than 2 million people by deploying solar panels over the state's aqueducts. Amazing, because I guess the water's evaporating in the hot aqueduct, and my goodness, capture the energy, get the water where it needs to go. A couple other fun facts. Number of University of California Nobel Prize winners is 70, which really exceeds that of virtually every other country in the world. It's an amazing thing. UC researchers submit nearly 5 new inventions per day in such diverse areas as agriculture, technology, biotech, and clean energy. Another fun fact, the University of California is awarded more NIH and NSF funding than any other institution in the country. Terrific. I will return a little bit later to help Provost Brown with the awards, but right now we have a wonderful treat for everyone. To take us through what's next, I will now turn things over to Yvette Gulat, Vice President for Graduate and Undergraduate Affairs and Vice Provost for Equity, Diversity, and Inclusion. So over to you, Yvette.
Great. Thank you, President Drake. Wow. Just wow. Now, was anyone expecting for Geoff to say friend A was Justin?
But this has been amazing and so inspiring. And it's incredible to hear about the impact that our students' research has and on the grand challenges that our world faces. So thank you to everyone who competed today, and definitely you are all winners. But now we have the opportunity to learn and hear from someone who knows firsthand about that impact of that research. And that is the very distinguished and highly regarded Regent Jose Hernandez. So for those of you who watched the full competition, you had the opportunity to hear his introduction earlier, and there is the introduction in your program. But I would like to say a few words about some of your many accomplishments, if that's okay. Regent Hernandez is president and CEO of Tierra Luna Engineering, LLC, an aerospace engineering consulting company. He also worked at the Lawrence Livermore National Lab for 17 years, impacting various critical areas, including co-developing the first full-field digital mammography system for earlier detection of breast cancer. On top of all that, he has been an astronaut on the STS-128 NASA Space Shuttle mission to the International Space Station. And of course, he actually just had the very difficult job of judging today's competition as part of our esteemed panel. So thank you, Regent Hernandez, for this opportunity to discuss a little bit more with you about the value you see in academic research.
Well, first of all, thank you very much for that kind introduction. And absolutely, research is the heartbeat of any economy of a country, because that's what keeps us competitive. And so by having a viable research programs at our 10 campuses, I mean, that we're doing our part in our country to keep us competitive. And judging by the talent that we just saw today, you can see how impressive it is that our programs are across all 10 campuses, because every one of these students did an outstanding job.
Absolutely.
It was very hard to grade them, and I just want to commend them for the excellent work, excellent skills they have at presenting, and the clarity they have in presenting their research. So a big round of applause to you guys.
So as we've all just seen, Grad Slam is all about taking complex ideas, complex processes, and sometimes mountains of data to convey meaning and value in accessible ways and in 3 minutes or less. So let's modify our Grad Slam challenge a little bit. Would you describe who you are in 3 words or less, and then tell us a bit about how you became you?
Passionate about science.
Very good.
Passionate about science. And then the second is describe about myself.
How you became you?
Well, I think I became me I'm a product of influence from my family and influence from society. I grew up as a migrant farm worker, first generation. Spent 9 months here in California, 3 months in Mexico, and followed the harvest of the crop across California. And seeing how my parents dedicated themselves and sacrificed themselves to give us an education, the only way I could see paying them back is to reciprocate and get that education. I went to graduate school, and in deciding what I wanted to be, that was very simple. If you can imagine a 10-year-old kid holding on to rabbit ear antennas, you kids don't know what rabbit ear antennas are, but on a black and white TV, as a 10-year-old, watching the very last Apollo mission, Apollo 17, astronaut Gene Cernan walking on the moon. I mean, that's when I decided I want to be an astronaut. And it was probably through osmosis, because I was hanging on to the antenna, that I got programmed to become an astronaut, but I never looked back, never looked back. And I tell you, what I subscribe to in life is a simple recipe that my father gave me, who only has a 3rd grade education. When I told him that night I want to be an astronaut, December 1972, he had the wisdom, this man with a 3rd grade education, to sit me on the kitchen table and give me this 5-ingredient recipe. He said, you really want to be what you want to be, follow these 5 steps. He says, define your purpose in life. Recognize how far you are. Draw yourself a roadmap so you know how to get there. Prepare yourself according to the challenge you chose. So you're going to college, he told me. And then he pointed outside, said, that effort you put out weekends and 7 days a week during the summer picking fruits and vegetables with your siblings, your mom and your dad? I said, yeah. He pointed to my books and says, you put that effort there. And so those were the 5 ingredients. As you mix that up, that's the recipe to succeed. I add the 6th ingredient, and I want to tell this to these kids, is the 6th ingredient is perseverance. Because I'm here to tell you, I fulfilled my dream of becoming an astronaut, but NASA rejected me not once, not twice, or 3 times, 11 rejections. Wow. It wasn't until the 12th time. So don't ever, ever give up on your dream.
Let's pick up on that thread a little bit about failure, because failure is such an important part of research.
Sure.
I wonder if people have an understanding of what it takes to get to those kinds of achievements. So talk a little bit about failure and the role of failure in research.
Well, no one likes failure. No one likes rejection. But let's face it, it's part of life and we have to deal with it. In my particular case, when I got rejected, the first time I got rejected, I remember I got a letter from NASA with NASA letterhead, and didn't even have my name. It said, "Dear applicant, even though you meet all requirements to become an astronaut, we regret to inform you that more than 12,000 people apply, and we could only hire 10 to 12 astronauts, and you're not one of them." But at the very end, it said, "Feel free to reapply." And that first letter, even though it didn't have my name on it, I framed it, and I said, "Hey, NASA thinks I could be an astronaut. It says so here." But then I got the 2nd, the 3rd, the 4th, the 5th, and the 6th rejection. And that's when that 6th rejection I didn't frame. I crunched it up into a little ball and threw it in the garbage. And I missed the garbage can, which is why I'm not a basketball player. And luckily, my wife was cleaning up and she saw the little paper and she saw that, read it. And so she knew I was giving up because she saw the letter all scrunched up. And she came to me and said, "What's this?" And I said, "Well, I'm giving up being an astronaut with 6 rejections." And she said, "You're not a quitter." I said, "You shouldn't quit because I know you. If you quit, that little worm inside of you is going to be gnawing at you and you're going to be wondering what if. So you should take this to the very end, because look at the end of the letter she read to me. Feel free to reapply next year." It says, "NASA's not kicking you out. You're kicking yourself out." And that's when I realized I got to apply. And that's when I did 3 different things to get selected that day, different from the others. The first thing is, it's a 3-element strategy. The first thing is make sure you have the minimum requirements to do the job, which I fulfilled and I did, given the letters that I got. Second is compare yourself to people that you want to be like. And so I compared myself to the astronauts, and they were pilots, scuba divers. I wasn't a pilot or scuba diver rated, so guess what? I became a pilot and I got scuba diver rated. And then the third thing is do things that differentiate you from the competition. And that's when I accepted that job at Lawrence Livermore, nuclear nonproliferation, which took me to Russia. No one wanted to do it. But guess what? I read in the newspaper that Russia and the US signed an agreement to build what was going to be the International Space Station. So it doesn't take a rocket scientist to figure out, even though I am one, that we were going to be working with the Russians up in space. So I put 2 and 2 together and I said, I'll take that job, because now I can go to Russia, learn the culture, learn the way of business, and more importantly, have a Russian language instructor and learn Russian. And then that's when NASA started to call me. So those are the things that the way I handled rejection is I said, okay, how can I do things better? How can I invest in myself to put me in a better position to succeed? And that's what we all have to do.
And everything about your career, yes, has been about making things better for everyone. And we've heard today the research that's been presented here is about making things better for everyone. So why should research matter to everyone? Why should we care about the kinds of science that's being done from your perspective? Why does all of this matter?
It matters because it improves the quality of life that we are living. Anything that helps us be able to find the cure for cancer, diabetes, insomnia or whatever we have with our circadian rhythm. All those things, we need to study them and understand them so that we can make our life better. I mentioned, a lot of people ask me what's my most proudest professional moment, and people expect me to say, well, I went to space, I was up there. But no, it was inventing that device that fulfilled digital mammography system. Now, that was important because the way that project got started, and this is where adversity, you can take advantage of adversity and it creates opportunities. The X-ray laser program that I worked in got canceled. And I was doing a lot of 3D Monte Carlo X-ray transport analysis. And so I had developed some good tools when that project got canceled. And so my boss and I felt we had an answer that we didn't know what the question was. And so when the project got canceled, instead of looking for another job, we said, let's use these tools and let's find that question. And the question was, how do you build a better mammography system for the earlier detection of breast cancer? And that's where the full field digital mammography system was born. That project was born. And now, we have images that are more clear, more information content. So now and everybody knows that earlier detection saves lives. And we opened up a whole new field of study called computer-aided diagnosis, where the computer helps you diagnose early precursors to cancer. So I'm very proud of that because I know we've saved thousands, if not hundreds of thousands, if not millions of lives with that project.
That is remarkable. That is just remarkable. You mentioned your father earlier, and one of my favorite moments is actually a TV commercial where you talk to your father, and it's about one of my favorite libations, but we won't say it here. But it's a great commercial. What does your father know about what you do, and what does he say now that you have achieved this journey?
Well, obviously, my parents were going to be very proud. There's 4 of us if we finished high school, which we did, if we went to college, which we all did. And of course, working at Lawrence Livermore Lab, he was very proud. And then becoming an astronaut, it's even more proud. But I tell you, I've come full circle in life. There's a saying, you could take a kid out of the farm, but you can't take the farm out of the kid. So when I came back home after leaving NASA and started my small consulting company, I also bought a small vineyard. And I knew how to pick grapes as a kid, but I didn't know how to operate a vineyard. And my dad, of course, does. And he's all of 84, and he still helps me. He gets on the tractor like I do. And he has taught me, when it's time to fertilize, irrigate, when it's time to harvest, all those type of things. So coming full circle, I spent a lot of time with him, and I think he's, in his heart, I think he's more proud that I own a vineyard than having gone to space.
There's always something to learn, isn't
Yes.
there? If you could wave your magic wand over all of California, right? So imagine you could just everywhere. What education investments would you make and why?
Well, I think if I could wave my wand, I would bring Bill and Melinda Gates here and tell them to invest in California in getting our kids interested in STEM education, and increasing the STEM pipeline into our schools, particularly into the UC system, because technology is what fuels our economy. And like these great students are doing today, they are the future. They are the present because they're doing work already, very important, but also the bright future of California and our nation. And so I would tell them, invest more heavily in outreach, invest more heavily in increasing that pipeline, because they have great foundations that get McKinsey Scott out here also, all these folks that are able to give, because the state and the UC system, I think, are doing their part, but we need to get more community investment into our backyards.
Very nice. Give us your best astronaut joke as a close.
Our best what?
Your best astronaut joke. Take us off stage with your best astronaut joke.
The best astronaut joke is probably more of an experience of being an astronaut. And that is you trained a lot to be an astronaut, you repeat things on the ground, you're training. For a 14-day mission, you train 18 months. So there's a lot of repetition. And I remember there was one particular activity that really frustrated me because it was replacing the air cabin filter. And the air cabin filter is just a panel of 4 screws, and you unscrew them. The screws don't even come out because they're interlocked, so they don't come out, so you can't even lose them. So you could unscrew it, remove the filter, and put the filter back in. And every time our trainers always used to insist, you got to do this. You got to do it. Come on, it's four screws. That anybody can do that, right? So anyway, fast forward, I go to space, and the first person whose activity is to replace the cabin air filter, yours truly. So there I go. I start, of course, you're floating at this point, right? Because you're going around the world once every 90 minutes at 17,500 miles an hour. So there I go, and we had an old saying, just like training when you do things up there in space. So I went in and I went to the tool thing and got the screwdrivers, just like training, Phillips screwdriver, good. And so then I float over and hover over the panel. And so there I proceed to unscrew. But what does physics tell you? For every action, there's an equal and opposite reaction. So as I torqued one way, my body torqued the other way, and the screw did not unscrew. And here I am, like scratching my head, looking around, embarrassed, making sure my colleagues didn't see, because I can't even unscrew a screw in space. What am I doing up here? And I'm scratched. And then I looked and I saw some gray tape on the bottom of what is the floor in our ground trainers. And so I went down there and I undid it, and sure enough, there were foot loops that you're supposed to put in. So you put in the foot loops, and now you're anchored. So now you could undo the screws, right? and so I did it. And then when I came back home, I said, man, I'm gonna get those trainers. They don't tell us, they're not training us right, and all that. So I go and I said, I bet you those loops aren't even there. And I went and where I trained dozens of times, sure enough, they were there.
Just like training.
But then I tell them, well, why don't you guys tell us? And they just smile because we always assign the rookie this first job. So, and we get him all the time. So that's what happened. But that was the best joke.
That's a good experience. Well, thank you, Regent Hernandez.
You're welcome.
Thank you for sharing.
Thank you very much.
And now we will turn the program over to Provost Michael Brown, who will preside over the award ceremony. Over to you, Provost Brown.
All right, Michael Brown.
Hello, everyone. I said to Vice President Yvette Galat, she has another career ahead of her. That's the next Johnny Carson, that's for sure. But I also want to say, it turns out that Regent Hernandez and I are connected on LinkedIn, and sponsor of this event. And but boy, the more I learn about you, the more fascinating your life is. So thank you for sharing. That was wonderful. Can we give them another applause for very illuminating? I also want to add my thanks to all the graduate student competitors for their outstanding contributions. And let me also say, your work helps, it helps so many understand the value of graduate education and the value of what a research university brings to the world. And look at what you bring to the world. I mean, regardless of what turns out on these awards, the world is your oyster. Y'all are awesome. So now this is the portion of the program where I become very popular. And everyone wants to hear from me because I'm here to announce the winners of this year's UC Grad Slam competition, a marvelous competition. How long have we been doing this competition? 2015 was the first year, 2016 at LinkedIn. It's been marvelous. But before I announce the winners, I'd like to take a moment, say I got your attention. I'd like to take the moment to acknowledge the graduate deans on each campus. I know you love your graduate deans. And these are individuals responsible for leadership on behalf of graduate education throughout UC. They helped make this day possible. And each of them had coordinated a successful competition at the local campuses, and that has resulted in our campus champions. So they not only host this event, but they commit to doing this annual tournament as well. So, and they are, I don't know if they're gonna picture them. We have Lisa Garcia Bedoya from UC Berkeley. Oh, no pictures? Okay. Jean-Pierre Deplanque from UC Davis. Yeah, you can applaud them. You can applaud them all, even if it's not at your campus. Okay. You know, we love them all. Jillian Hayes at UC Irvine. Susan Etner at UCLA. Chris Kello at UC Merced. Sean Bowler at UC Riverside. James Sautal Anthony at UC San Diego. Niket Blake at UC San Francisco. Lila Roop at UC Santa Barbara. And Peterbilt at UC Santa Cruz. They work hard on those campuses on behalf of graduate students. I wish it was easier than the competition, and the competitions are hard. But I do also want to thank again our sponsor, Thermo Fisher, one of our other sponsors, because they are providing the cash awards today. So yeah, let's give a hand to Thermo Fisher. Okay, now we will announce the People's Choice Award, the third place award, the second place award, and the Slammy. You've heard of the Grammy. This is the Slammy. Do we have have it to show? Oh yeah, we you got it. This is the Slammy, a highly coveted award. Last year you wrote that in my script. All right. Now, all of the presenters were extremely impressive, so we know this was very difficult for the judges. But let's hear from our audience judges first, and our thanks to everyone who took the time to cast their online vote. Okay, we can now announce the system-wide People's Choice Award. I know I'm getting breathless here too. The winner will receive $1,000. And I want to thank everyone who has cast their award. Why do you do this? Okay. Okay. Now the award goes to, yeah. Wei Gordon, UC San Francisco. There we go. Niket Blake is the graduate dean. Niket Blake. Oh yeah, take them pictures. Now we have the third place award. And this award goes to I'm supposed to open these things. Rachel Sousa, UC Irvine. Jillian Hayes, the Dean. Oh, let me get out of that picture. We have the second place award, and it goes to Amanda Quirk, UC Santa Cruz, Peter Beal, Grad D. That is awesome. It's exciting. It's time to name our winner. But, no. If this is a reality TV show, we'd be cutting for a break. We say, come back next week, same bat time, same bat channel. We'd have to wait to see who got the rose, but that's not us. So there's only one thing left for me to do. It is my pleasure to announce the UC system-wide Grand Slam champion, and the Slammy goes to Isn't that wonderful? Oh, I know I'm delaying things. Okay. To Justin Lee, UC Berkeley. Lisa Garcia Bedoya, the graduate dean. Okay. This is just awesome. There is actually one more award. This is an annual competition, and we have a perpetual plaque. And on this perpetual plaque, we have the engraved names of our deans who have won this competition. Well, they're students, but you know what I mean. All right. So and so the Dean Lisa Garcia Bedoya. Is this? It ain't telephone. Well, anyway, she will be on this plaque in perpetuity here. So and let me finish up. Her name will be added. She has bragging rights for 364 days. Okay. And on behalf of President Drake and all of us at the Office of the President, we want to thank our partner, LinkedIn, for hosting this Grad Slam event here in their San Francisco location. Beautiful headquarters here. We have missed you during the pandemic. We're glad to be back. We thank our fabulous award sponsor, Thermo Fisher, once again, for their contributions. They have been very generous in supporting the full cost of prizes for our student winners. Thank you, thank you, thank you. We appreciate the work done by so many at this event, the creativity that goes into it, the time, the talent, the effort. Um, uh, and I want to thank the UCOP Grad Slam team. Can y'all just stand? Let- All of y'all that were involved, that's right, in helping to put this event together. Thank you so much. Um, uh, Grad Studies and our marketing comm team are so intimate to this event. And finally, we want to thank each of you for joining us at this special event. We appreciate your wonderful support. We appreciate our judges so very much for your time and effort. We have given you just a glimpse of what UC research is achieving. And the beautiful thing about research, it just begets more research. It begets more studies. It begets more discoveries. Um, uh, we need to continue to fund it. We need to continue to advocate for it, and we need to continue to engage others with it. And this is just one of our efforts in doing so. So thank you all so very much. Please tune in next year. It's going to even be greater next year. So thank you very much.